Hello, I'm David Schaffer, a professor of chemical and biomolecular engineering, bioengineering, and molecular and cell biology at the University of California, Berkeley, where I'm also director of QB3 and Bakar Labs. So I'm thrilled to be able to share some of our work with you today, where I'm going to be discussing directed evolution of AAV delivery systems for clinical gene therapy. The motivation for all of our work is really shown on this slide here, where regardless of which tissue you look at, there are a number of conditions, both inherited as well as more complex or idiopathic, that involve long-term progressive degeneration of a tissue, loss of cells within that tissue, progressive decline in function, and loss of quality of life for patients with these conditions. So there's very clearly a need to be able to develop new therapeutics to treat these conditions. So if we back up all the way to the central dogma of biology, a very basic tenet within that field, it posits that information flows from DNA, which is the long-term repository for storing information within our cells, through RNA and then to protein. And from the point of view of a drug developer, each one of these levels of molecules is a potential drug target. So the majority of therapeutics or pharmaceuticals used in the market and in the clinic these days target at the level of proteins. Small molecules float around, typically flow through membranes inside of cells and bind to the active site of an enzyme in order to modulate or inhibit its function. Monoclonal antibodies float around on the outside of the cell and typically do the same by binding to a receptor or to a ligand and disrupting their interactions. We know it's also possible to drug at the level of RNA. There's RNA interference, antisense oligonucleotides, and of course, thanks to Pfizer and Moderna, we know that mRNA itself can be a therapeutic. I've always gravitated towards drugging at the level of DNA. So if you add new DNA to a genome, that's a gene therapy. If you change the sequence of existing DNA, that's a genome edit. If you add a brand new genome to a tissue, that's a cell therapy. And the advantage of being able to drug at the level of DNA is that unlike small molecules, proteins, and RNA molecules, which have a limited pharmacokinetic half-life, DNA can persist within a patient through the rest of their life. So you can begin to think about treating the underlying mechanism of a disease by treating the gene that causes that disease, and furthermore, doing so in a manner that is one and done, single administration, long-term therapeutic benefit. The big challenge with this field, however, especially for gene therapy, which is going to be my focus today, is delivery. That from the point of view of a cell, DNA is a very, very large molecule. It's charged, it's not particularly stable, floating around in serum, and it's very difficult to get it past cells' membranes and into the nucleus, where it can actually supplement the genome or add to the genome of a cell. So to do so, we need a delivery vehicle, and many or most delivery vehicles that are under clinical development these days are based upon viruses, because of course viruses often deliver nucleic acid or DNA specifically as part of their replication cycle. So we can take a harmless virus, such as the one shown here, adeno-associated virus, never associated with a human disease, it's not a pathogen, and essentially re-engineer it or trick it into delivering a therapeutic piece of DNA rather than its own genome. So AAV is an interesting virus in that it's among the smallest of all viruses, only around 4.7 kilobases of information. And in addition, it's the smallest among the smallest of particles, around 26 nanometers in diameter. And the way that it works is that it has several open reading frames. The first set of ORFs are called rep, and these encode 4 enzymes that mediate the replication of the viral DNA and assist with the packaging of the viral DNA into the capsid. The second ORF that I'd like to call attention to is CAP, which encodes 3 capsid proteins, 60 copies of which self-assemble to form the icosahedral structures you see on the right-hand side. Once the icosahedron is assembled, a rep protein docks at a pore present at the 5-fold axis of symmetry, grabs a single-stranded genome of the virus, and pumps that genome into the interior of the particle to be able to create an infectious virus. So another interesting feature of this virus is that a number of natural variants have been isolated, over 100 of them, and they contain diversity, especially in the surface-exposed amino acids on the viral capsid. So this results in some variability in the delivery properties of the virus, raising the possibility that by engineering the surface amino acids, you could actually create a targeted or more efficient or redirected gene delivery vehicle. So the way that you convert AAV into a recombinant gene therapy is by simply cutting out the viral genes from the genome, rep and cap shown here, placed onto a separate piece of DNA, which then liberates space for the insertion of a gene of interest inside those viral elements, those ITRs shown in the upper left-hand part, And it's those ITRs that specify which DNA go inside of the variant. And as a result, the gene of interest is packaged inside the virus rather than the virus's own DNA. So the resulting virus delivers that gene of interest, and it has a number of features that are advantageous. So it's relatively safe. 90% of the human population has been exposed to one or more variants of AAV and never even noticed it. It's somewhat efficient in gene delivery clinical trials involving delivery typically to tissues like muscle, liver, lung, brain, and retina. Upon depositing the viral DNA into the nucleus of target cells, that DNA essentially establishes a latent infection where it can persist for long periods of time, years or even decades. There has been a lot of progress in manufacturing these viruses, so that it is manufacturable to clinical and in some cases commercial scale, though there does still need to be significant improvement there, which I'll get to a little bit later in the talk. And apologies for this being a little bit out of date, but there are actually now 7 FDA-approved gene therapies for a variety of conditions, ranging from spinal muscular atrophy to Leber's congenital amaurosis type 2 to hemophilia A and B. So there are a number of challenges that remain to be overcome within the field, however, and a lot of those challenges arise from the fact that this is a complicated mode of therapeutics. So first of all, we have to manufacture the virus. This is a 4.5 to 5 million molecular weight particle composed of a complicated assembly of proteins with nucleic acid on the inside. It's typically manufactured in an adherent mammalian cell process where you have to transfect plasmids into cells at scale and then isolate the virus a few days later. Upon isolating and doing the downstream processing for the virus is administered, has to be able to home to a tissue or organ of interest, such as the heart after an intravascular injection, has to be able to extravasate out of the vasculature, penetrate the parenchyma of the tissue, find a target cell by engaging with the receptor on the surface of that cell, undergo endocytosis. The virus has a mechanism to escape endosomes. It has nuclear localization activity that enables it to traffic to the nucleus. And then finally, once inside the nucleus, that capsid has to disassemble in order to release the actual active ingredient of the therapeutic, which ends up being ultimately a double-stranded DNA molecule that contains typically a promoter, a coding region, as well as other regulatory elements, including polydenylation sequences. So that drug product itself is also very complex. So there's several challenges associated with this complexity, and the first of these is the delivery challenge. And there's an old joke in the gene therapy field that there are only 3 problems in gene therapy: delivery, delivery, and delivery. The second is a manufacturing challenge where it's difficult to produce this product at scale. By creating a more efficient delivery vehicle, you can potentially reduce the dosage necessary for administration. So solving or addressing the delivery problem also helps out with the manufacturing challenge, but we also need improvements in both upstream as well as downstream manufacturing to address this second category. And then finally, although I'm not going to be talking about this today, we have a regulation challenge, which is that the first gene therapies that were approved typically used constitutive promoters that produced or expressed that transgene in every cell where the virus ended up landing. So as time goes on, likely we're going to treat conditions that are more complex, where it's important, where we control when, where, and how much of a therapeutic RNA or protein molecule is produced. And as a result, we're going to need to be able to get more complex regulation to address that challenge. But I'd like to focus first on delivery and then second on manufacturing. So why is there a delivery problem? Typically, the gene therapies that have been used to date are based upon natural versions of AAV, and these are inefficient and untargeted. So they have a number of reasons for this. First of all, as I alluded to, greater than 90% of the human population has been exposed to one or more natural versions of AAV, which means that we typically have high titers of neutralizing antibodies that would end up eliminating the natural versions of AAV that might be administered to body fluids that have antibodies, such as serum, vitreous fluid, lung fluid, etc. AAV is inefficiently delivered to most tissues inside the body. It, when applied intravascularly, for example, it typically is taken up within the liver and degraded. It's not particularly great at delivery to the liver, but certainly it's not effective at delivery to other tissues in the body beyond the liver. It typically inefficiently spreads within a given target tissue, has zero ability to target delivery to specific cells within that tissue, is inefficiently taken up into those target cells and is challenging to manufacture at high scale. So this begs the question, how is it that we actually have 7 FDA-approved products to date? So the answer to that is that for these products, the delivery challenge is typically brute forced in one of 2 ways. So first, one can administer extremely high doses to be able to overcome the inefficient delivery for a given particle. And the second is that some of these treatments involve invasive routes of administration to place the virus right next to the target cell, because otherwise the virus wouldn't have a prayer of reaching that target. So there are approaches, there have been treatments in which investigators have tried to compensate for the low delivery efficiency by really dramatically increasing the dosage. And then you run into a problem that while AAV is a relatively stealthy and non-immunogenic virus, if you increase the dose high enough, the immune system begins to take notice. So for example, there was a clinical trial in which Astellas was trying to use AAV8 to deliver a therapeutic payload to muscle inside the body, skeletal muscle actually. And AAV8, by and large, localizes to liver. So perhaps for every 1,000 genomes or every 1,000 particles that make it to the liver, only one makes it to the heart. To compensate for this, they dramatically increased the dose up to the level of over 100 AAV particles for every single cell within the body, or 65 milligrams of AAV. This led to extreme levels of localization in the liver, and an immune response against capsid antigens actually resulted in liver toxicity that ended up resulting in the loss of several patients within that clinical trial, which was then discontinued. So the bottom line is that viruses never evolved in nature for our convenience to use as therapeutics, and so it's up to us to re-optimize them at the molecular level to be able to adapt them for the applications we're trying to use for them, rather than be at the mercy of whatever nature happens to have thrown over the fence in the form of natural versions of these viruses, which are inefficient and untargeted. So we'd like to re-engineer this particle, but again, this is a really complicated process. This is a 5 million molecular weight particle that is, again, composed of a complex assembly of protein and nucleic acid, and it's not very easy to engineer this compared to engineering the structure of a small molecule. So a number of years ago, when I started my lab back in 1999, we began to take notice of emerging technologies in protein engineering and began to apply this towards gene therapy. So specifically, there was an emerging approach called directed evolution that came out in the early 1990s and was initially applied towards engineering enzymes, and was eventually awarded the Nobel Prize in Chemistry in 2018. So I started my lab in the late 90s, and we began to adapt that approach for the first time to be able to create improved versions of AAV viruses for gene therapy. So the way that technology works is that you take DNA encoding a natural version of the virus, and specifically the capsid, because it's that protein shell that serves as the delivery vehicle for the nucleic acid. We then use a variety of approaches to genetically diversify that capsid to be able to create a large gene pool. That gene pool is then packaged into a library of viral particles, where every single particle is composed of a protein shell surrounding the DNA that encodes that protein shell. So you can think of it as each particle being barcoded with its own genome, its own DNA. And the size of these libraries is likely in the billions. We've not been able to do enough sequencing to fully capture the size of the library, but it's likely it exceeds 1 billion. So then perhaps, for example, this variant shown here with the red around it in the right center part of the panel is the variant that would be ideal for delivery to the heart after an intravenous administration. So we can then take that entire library within a syringe, inject it intravenously into an organism. We started out with small organisms and then progressed towards large organisms to maximize the chances that these particles could translate to success in a human gene therapy clinical trial. And through successive rounds of selection, where we inject the virus, isolate the target cells, amplify the virus that made it there, re-administer it into the next round of selection, we then, in Darwinian fashion, converge that large library into the fittest variants, the ones that are most fit or most suitable for the gene therapy task we're asking them to perform. So now that I've introduced the concept, I'd like to progress through a couple of bullet points in which we initially applied it as a proof of concept into a mouse, then progressed into the clinic, and then have been using that approach to be able to address other problems in gene therapy, namely manufacturing. So the first target tissue that we began to work with in vivo was the retina. And the structure of your retina is shown here, where at the back of the eye, on the surface area of the retina, we have a number of layers of neurons. Shown at the bottom is the ganglion cell layer, the GCL. All the way at the back of the eye is the outer nuclear layer, which contains the photoreceptors. And unfortunately, mutations in over 200 genes, as well as more complex conditions like macular degeneration, can kill off photoreceptor neurons, progressively robbing a patient of vision. So you'd really like to deliver a DNA medicine to those photoreceptors to be able to treat that condition. And the most straightforward way to administer a therapeutic into the eye is what's called an intravitreal injection, where you take a needle, do a local anesthesia, poke the needle through the white part of the eye, and inject the fluid containing the therapeutic into the liquid humor of the inner eye. So while this is a little bit cringeworthy, this is actually done millions of times a year in the United States to administer therapeutics for the treatment of wet age-related macular degeneration. And the most prevalent product used in that field is called Eylea, or otherwise known as aflibercept. Remember that for later, please. And this is a protein that neutralizes vascular endothelial growth factor, which plays a role in the pathology of wet age-related macular degeneration. So investigators inject the protein inhibitor of VEGF, and this ends up neutralizing that protein and halts the progression of wet AMD for patients. So it makes sense to be able to administer AAV by the same route of administration. It would give the AAV the opportunity to come into contact with the full surface area of the retina, and it's a very safe and non-invasive route of administration. But the problem is that natural versions of AAV are incapable of penetrating through the couple hundred microns of dense tissue that lie between the fluid of the retina and the outer nuclear layer, the photoreceptor neurons. So to be able to get those photoreceptor neurons, investigators had to invent a new route of administration, which involves taking a needle, progressing it through the vitreous of the eye, the liquid humor of the eye, and poking the needle through the retina at the back of the eye, and injecting the fluid behind the photoreceptor layer, and as a result, in between the photoreceptors and the underlying retinal pigment epithelium. Then fluid is forced between those two layers, which presents a couple of problems. The first of these is that it detaches the photoreceptor layer from the underlying RPE. So this is by definition a retinal detachment. And so this ends up damaging the retina in some cases, especially in patients who already have an underlying retinal degenerative disorder. And the second problem is that it's difficult to control in what direction and how far away from the needle tip the fluid laterally flows underneath that photoreceptor layer. So typically this injection only reaches around 10% of the surface area of the retina and leaves the rest of the retina subject to disease progression. Yet this approach was good enough for an FDA approval. In fact, the very first FDA approval for an AAV gene therapy, which is currently commercialized by Roche. So we wanted to probe a different route of administration, which was to inject this library into the vitreous of the eye, wait a couple of weeks for the most successful variants to be able to reach the outer retina, the photoreceptor neurons, PCR out those variants, and then package that library again, re-administer them. And by doing this for 6 rounds with initially a mouse as a proof of concept, we isolated one particular variant that was dominant in the resulting selected pool, and that's shown here on the lower right-hand side, and it has some diversity in the surface of the capsid, some altered amino acid sequence, in a position that places it in a way that can alter the way that that virus interacts with cells and tissues within the body. So we wanted to test whether or not this resulted in improved gene delivery. And so we took either the parental virus known as AAV2. So shown here is an intravitreal administration of AAV2 into a mouse. You can see if you inject in the vitreous, which is all the way at the bottom of that image, barely any of it reaches into the retina. And AAV2 is the variant that's used in that Roche product to treat a blinding condition. And you can see why they had to do a subretinal surgery, because otherwise the virus doesn't penetrate into the retina. The variant we selected, by contrast, is able to reach very deep into the retina and very efficiently deliver DNA to a broad range of cell types, including photoreceptors and retinal pigment epithelium. So this work was done in a mouse, and there's a saying within the field of evolution that nature teaches you, you get what you select for. So this variant worked well within a mouse, but didn't work well within some of the larger animal models that we wanted to test it in prior to heading into human clinical development. So our next goal was to take the success within the mouse, which shows very efficient delivery deep into the retina and across the surface area of the retina, and see if we can achieve that within a large animal model, initially a non-human primate, and then ultimately a human primate. So to do so, I founded a company called 4D Molecular Therapeutics in 2013. We took this company public in 2020, which simply meant that we had the capital to be able to take the technology into clinical development. And currently, the company is developing drugs in the retina as well as in the lung. And the lead product is for wet age-related macular degeneration. So we redid the evolution within a non-human primate, arrived at a different version of the virus, and shown in the series of images is the delivery properties of that engineered version. In the bottom center is a negative control with AAV2 injected subretinally. And you can see that there's kind of a random shape of DNA delivery shown within that dotted blue square. Again, indicating you can't really control where the DNA goes. On the bottom left-hand side shows what happens if you take AAV2 and inject it into the fluid of the eye. For reasons I won't go into due to time, it only delivers DNA to a small population of retinal ganglion cells, but doesn't reach any of the photoreceptor neurons. This is the engineered version that we created. Shown on the right-hand side is in-life images of a non-human primate, showing that by 3 weeks, there's very widespread gene expression. We ended up testing the retina and doing a lot of microscopy images to quantify how good the delivery was to photoreceptor neurons. And across the entire surface area of the eye, as you can see in that panel that says multiple layers of the retina to the left, there's very strong delivery to photoreceptor neurons. So we decided to develop several gene therapies based upon this virus. And the lead product, as I mentioned right now, is wet age-related macular degeneration. So you'll remember I mentioned earlier that the most prevalent protein therapeutic used for wet AMD is called Eylea. And this is injected into the eye to be able to neutralize VEGF, which plays a pathogenic role within wet AMD. The problem with that protein therapeutic is that proteins have a limited half-life inside the body, so patients have to get something on the order of 8 to 12 injections per year. So it's fine to get poked in the eye once, but if a patient ends up having to get 10 injections a year, typically compliance begins to go down, patients skip injections, and wet AMD then unfortunately progresses within these patients. So what we did quite simply was to take DNA encoding Eylea or aflibercept and encode it or embed it into the virus, so that the virus would then deliver DNA encoding this therapeutic. Cells within the eye would secrete it as little biofactories, and as a result, the eye would provide its own steady supply of this protein therapeutic, and then hopefully patients would see a significant reduction in the need for protein injections. Shown here is the result of a 6-month in-life study within non-human primates. This is a vascularization model that simulates wet AMD. And shown here on the in this center data panel, on the y-axis is the concentration of aflibercept. On the x-axis is the dosage of the gene therapy administered to this non-human primate. And you can see even at a dose of 10 to the 11th, it turns out there are therapeutic levels of aflibercept that are produced within the eye. Next, we wanted to test whether or not this aflibercept could actually reduce the incidence of vascularization within this model of wet AMD. And you can see that as compared to a vehicle control on the right-hand side, even a dosage of 10 to the 11th was sufficient to completely inhibit or block the formation of these vascular lesions. So based on this, the study progressed into a human clinical trial sponsored by the company. And these are some data that were presented initially publicly about a year ago or so. So in a phase 2 human clinical trial, in a cohort of 20 patients, they received a dosage of 3 times 10 to the 10th vector genomes per eye, which is actually lower than the lowest dose that had been previously administered to non-human primate. These patients typically had received 10 protein injections per year. And importantly, there was no significant clinical intraocular inflammation reported at all, indicating it was a safe product. In this bottom panel, you can see on the left-hand side, each one of those colored circles represents an injection of the protein. Each row is a single patient. So there are 20 rows from top to bottom to represent the 20 patients. And you can see with the introduction of the gene therapy, shown by the vertical line towards the right of that panel, there's a substantial drop in the need for protein injections. So there was actually a 89% a reduction in the need for protein injections, and 2/3 of the patients were injection-free on an annualized basis within this initial data report. So this indicates that directed evolution can create a virus that at very low dosage can result in a therapeutic effect in human. And that dosage is small enough to actually make this not very challenging to manufacture. I'd like to finish the last several minutes by talking about the application of this genetic diversification and functional selection approach to directly take on manufacturing of AAV. And the need for this is that while the ocular gene therapy I mentioned to you just a second ago for wet AMD, it's a relatively small tissue, and we're able to get the virus efficient enough to be effective at a very small dose. And as a result, manufacturing is not a significant burden for that therapeutic. But if you consider other conditions where the dosage required is substantially larger, all of a sudden manufacturing becomes a significant challenge. So a single bioreactor run for wet AMD, for AAV rather, generates on the order of 10 to the 16th AAV molecules in the state-of-the-art bioreactor known as an iCELLis 500. So if you take a look at the bottom, there are gene therapies that are FDA approved for both spinal muscular atrophy, SMA, as well as for Duchenne muscular dystrophy, DMD. So the total surface area of tissue culture polystyrene or of cell culture required to manufacture AAV for SMA for the annual incidence is 20,000. So that's 40 bioreactor runs to be able to treat the annual incident population of SMA. So 40 bioreactor runs is feasible. But once you head to DMD, all of a sudden you're talking about 7.5 million square meters to be able to treat the prevalent patient population. So that becomes prohibitively difficult. And this was actually noticed a number of years ago in the New York Times that wrote an article indicating that viral manufacturing was actually becoming a roadblock within the field. So we began to propose the idea that just like natural versions of AAV weren't really engineered by nature to be therapeutics, so we wanted to improve upon those natural versions and make much more efficient targeted AAVs. By the same token, AAV manufacturing is typically conducted in adherent mammalian cell culture, and specifically HEC293 cells. And there's nothing about HEC293s that prepared them to be optimal for AAV manufacturing. So we decided to start using diversification and selection approaches to engineer new versions of 293s that were optimal, or at least better, for AAV production. So to be able to get the genetic diversification within the 293s, we began to use an approach called genome-wide CRISPR screening, building upon CRISPR technology, which was originally invented at UC Berkeley. So there's a way where you can take Cas9, the enzyme of the CRISPR system that is a nuclease and cuts a target site as guided by a guide RNA. And you can generate a nuclease-deficient version of Cas9 and then fuse it to transcriptional activation machinery, transcriptional activation domains. You can then take a guide RNA and direct that Cas9 to a specific location in the genome, such as a promoter for a gene. And that transcriptional activation machinery will upregulate expression of that gene. So that's a gain of function approach. And there's a corresponding loss of function approach, which I'm not going to focus on as much, but you can take a nuclease containing Cas9 or active Cas9 and use a guide RNA to direct it to, let's say, the first intron or first exon of a gene. And as a result, this creates an indel that knocks out that gene, throws it out of reading frame. And because this can be done with guide RNAs, you can do it in a genome-wide fashion. So you can have a library of guide RNAs, which will direct this machinery to, in principle, any gene in the human genome. So what we did was we took those guide RNA libraries and put them inside of an AAV genome. So specifically, the transcriptional activation machinery guide RNA library had around 70,000 variants, or 3 genes targeting each of 23,000 genes within the human genome. And we packaged this library into AAV. So every color here shown on the right indicates not a different capsid structure like it did during the first part of the talk, but a different guide RNA inside that viral genome. We can then take that library of AAVs, infect 293 cells, and importantly, within these 293s, we've engineered them to overexpress the Cas9 activation machinery. So we wait 48 hours for the AAV to unpackage, where there's one or no AAVs per cell. It begins to express the guide RNA. The guide RNA directs the Cas9 to a particular gene within that cell. It upregulates that gene. And if that is a gene that actually enhances AAV packaging, we can sense that. And the way that we sense that is to take the other packaging constructs for AAV, transfect them into the cell population. And again, if that upregulation of a particular gene improves AAV production, then there should be more copies of that genome of AAV from that cell packaged into capsids. And again, to remind you, that genome contains the guide RNA. So that ends up enriching beneficial guide RNAs within that population. So we can then take that newly packaged selected library, reinfect the cell, and go through progressive rounds of selection so that we end up increasing the prevalence of guide RNAs that are favorable for AAV packaging. We can then use Illumina to sequence the guide RNAs and determine by what factor they were enriched during the selection process. So you can see here, again, focusing on the transcriptional activation screen, we had genes including things like ITP-RIP or SKA2, which I hadn't heard of before doing this screen. Again, that's the beauty of a non-biased screen. You're surprised. And ITP-RIP is an inositol triphosphate receptor interacting protein. SCA2 is spindle and kinetic core associated protein 2. And these, it turns out, play a role in calcium signaling inside of cells and in the process of mitosis. So we then tested the impact of these guide RNAs and then ultimately generated stable cell lines expressing or overexpressing cDNA and determined the impact on viral production, initially with AAV2. So you can see here, we end up with a 2 to 2.5-fold increase in viral titer. And that is genome titer, but that has a corresponding increase in infectious titer. And when we began to combinatorialize these two guide RNAs, or specifically these two genes, we see we're getting up to a 4-fold increase in AAV production. So this would correspond to a 4-fold decrease in the surface area necessary to manufacture AAV for a given patient. And fortunately, this genetically modified cell line also worked for other AAV serotypes, such as AAV6 and AAV9. So I'd like to conclude with a couple of bullet points that genetic diversification and selection are powerful tools that can be harnessed to solve problems in gene delivery systems. And we've used this approach to re-engineer AAVs, to reprogram them for enhanced efficiency, targeted delivery, as well as the capacity to evade pre-existing immune responses, pre-existing antibodies, in work I didn't discuss today. So the vectors we have engineered have been used in a number of human clinical trials and have resulted in positive data out of many of these that have reported data to date. And in addition, we've extended this genetic diversification and selection approach to other problems, specifically manufacturing, as well as in work I didn't discuss today, be able to address new approaches to solve the regulation challenge, that I alluded to at the beginning of my talk. So I'd like to acknowledge the people who actually did this work. Leah Byrne and Denise Dalcara developed the vector for delivery to the retina of mouse. These are a group of people within the company, not everybody, but a group of people who are responsible for creating the vector that went into human clinical trials in the retina. And in addition, the cell line engineering, or the genome-wide screens, were conducted by Chris Barnes and Hansho Lee. So thanks so much for your time and attention. I'd be happy to, in the future, address any questions.